Today we are going to look at a number of topics closely related to cell biology. The first topic we are going to look at is intercellular communications, or basically what we call cell-to-cell signaling. In this topic, we are going to address the following things. We are going to state how cells communicate with each other. We are also going to see the difference between direct and indirect types of intercellular communications. We are going to define some two terms here, one being what we call secretory cell, and the other one being what we call target cell. We are going to describe the mechanisms of signaling that occur at the target cell. We are also going to outline the classification of chemical messengers. And lastly, I'll give you examples of common chemical messengers, or what we call ligands. It's a short lecture though, in as much as the objectives appear a lot. So first, we need to understand that cells can communicate with each other. And that is what we call intercellular communication. Communication between one cell and another within the body is what we call intercellular communication. There are two types of intercellular communication. There is what we call direct type of intercellular communication. There's also what we call the indirect type of intercellular communication. So to begin with, when we talk of direct type of communication, this is how we call it when the two cells are physically in contact. So direct communications occur where the two cells are in physical contact. That therefore means that in the indirect type of communication, the two cells are not in contact. So when we talk about direct communication where the two cells are in contact, there are two possibilities that we can talk about. When the two cells are in contact, you want to know what is actually linking the two cells. So there's a scenario where the two cells could be linked by gap junctions, like what we see here. The two cells are linked by gap junctions. Gap junctions enable the cytoplasm of this cell and the cytoplasm of the other cell to be in continuous. That means, therefore, that if there is any ion here, like this molecule here, it can easily diffuse to the other one. You remember we talked about types of transport of small molecules. Those small molecules can easily go through the gap junctions to the other cell, and vice versa. If there is something that is higher concentration on this side, it can easily move to the other side. So cells do communicate via gap junctions. Communication through gap junctions is an example of direct intercellular communication. Examples of cells that communicate through gap junctions include smooth muscle cells and cardiac muscle cells. Adjacent cells will move, or rather will communicate through such junctions. And communication through gap junctions is usually very fast. The second type of direct intercellular communication is where the two cells are communicating via some surface molecules. For example, in this image, there are some surface molecules, which means that those molecules are on the cell membrane. The surface molecule of the cell and surface molecule of the other cell. And kind of this other surface molecule is a receptor for this one. So the two cells can communicate with each other via surface molecules. The best example of cells that do communicate through surface molecules would be some white blood cells, or generally, let's say, cells of the immune system. They tend to use surface molecules for communication, cells of the immune system. So that is direct communication. Okay, so people are saying they're not hearing me. Are the others hearing me?
Yes.
Yes.
Oh,
Yes.
so it's their network. Okay, let me just write that. All right, so we've talked about direct type of communications. Now let's talk about the indirect type of communications. So as I mentioned earlier, for direct communication, the cells are in physical contact. That means that for indirect communication, the cells are not in physical contact. The two cells are not physically linked. There is some distance between them. If there is some distance between the two cells, then you ask yourself, how will these two cells then communicate? Now, first of all, we need to understand that for two cells to communicate, one cell must initiate that communication and the other cell must respond to that communication for us to say that communication has occurred. For these two cells to communicate, yet there is physical distance between them, it means that the cell that initiates the communication will communicate using chemical messengers. And that's the whole point about indirect communication. For indirect communications, communication is made possible by release of chemical messengers. The cell that initiates communication releases chemical messengers. Those are the messengers, those are the chemicals that will move to the cell that responds to that communication. Now, the term given for chemical messengers is ligand. So a ligand is a chemical messenger. Now, let's define some terms therefore. The cell that initiates communication is the one we call the secretory cell, because it is the one that releases the chemical messenger. On the other hand, the cell that responds to the communication, which means it's responding to the chemical messengers which have been released, that particular cell is what we call the target cell. It is important, therefore, to note that the secretory cell is the one that synthesizes and releases the ligand. I gave you that assignment of protein synthesis. I hope you're done by now. So the secretory cell will synthesize that ligand. It could be a protein. Then releases that ligand out of the cell. Of course, usually through exocytosis. Then that chemical will travel through different mechanisms that we'll talk about later, maybe not today, okay, partially today. Those chemical messengers travel through different mechanisms until they reach the target cell. Now, what is unique about the target cell is that the target cell contains receptors. Those receptors could be on the cell membrane or could be within the cell itself, inside. Either way, the target cell contains receptors for that particular ligand. So the ligand could be a hormone, for example. It means that the secretory cell will release the hormone. Take for example, your pituitary gland producing, let's say, growth hormone. You want this growth hormone to stimulate some cells in your bone. Let's say it's in the femur. So the cells of your femur will then contain receptors for growth hormone. And so that growth hormone will actually target the bone cells. In this case, maybe they're in the femur. And then the target cell is the one that will then respond to that communication by initiating particular cellular responses. In this case, now you need cell division, and that's what will happen. So anyway, the point here is that the target cell contains receptors for the ligand. If a cell does not have a receptor, then it is not a target cell. Target cells are defined by the presence of receptors. If you remember, we talked about cell membrane proteins, and we said that one of the classes of cell membrane proteins are receptors. Now let's see what happens at the target cell once the chemical has reached the target cell. So what happens? Once the chemical reaches the target cell, the chemical will bind to its receptor, as I've just told you. The binding of the ligand to its receptor activates an intracellular molecular cascade. Now I want to just leave it at that because there's a lot of intracellular molecular cascade that happen, which is beyond the scope of these lectures. You will mention some of them in your pharmacology. You'll mention some of them in your immunology. But for now, let me just say that there are a lot of intracellular molecular cascade that are activated when the ligand binds to its receptor. Eventually, that molecular cascade will activate things within the cell. Now, what kind of things will be activated within the cell? There are some target factors within the cell that will be activated by this molecular cascade, and it depends on the type of communication that was there. So it could be that an enzyme is activated within the cell. It could be genes are activated within the cell. It could be the cytoskeleton is what is activated. You remember microtubules, microfilaments, intermediate filaments. These are the ones which are activated. Or it could be ion channels are the ones which are activated by this intercellular cascade. Now, depending on whatever is activated, then you can eventually have final cellular responses. For example, if you activate an enzyme, then you expect to have alteration in metabolism. Either there is increased metabolism, or you actually inhibit metabolism, depending on what type of enzyme you've activated. If it's gene that is activated, then it means that you'll have altered gene expression in terms of protein synthesis. If it is cytoskeleton that is activated, then it means you may have, let's say, movement of the cell, because cytoskeleton is responsible for movement, motility in the cell. One of their functions is movement. It could be cell division, because microtubules are important in cell division. If it is ion channels which are activated, then you may have transport of some molecules either into or out of the cell, because it means you open ion channels. And so it means that some small molecules can either enter or leave the cell. So this is what happens at the target cell. The ligand will bind to the receptor, activate intracellular molecular cascade, very extensive cascade. I don't want to go into it, but eventually you activate some targets within the cell. Could be enzymes, could be genes, could be cytoskeleton, could be ion channels. So depending on what has been activated, then you'll have final cellular responses. The cell will respond in a particular way. Right. Having said that, let me now give you classification of ligands. We have said that ligands are chemical messengers. Now we classify ligands in different categories. We can have what we call autocrine ligands, paracrine ligands, neurotransmitters, hormones, and neurohormones. These are five classes of ligands, and I want us to talk about each of them. So let me just move to the next slide, because the names are going to appear again. Let's begin with paracrine ligands or paracrine chemicals. What are we calling paracrine ligands? Now, you call that chemical paracrine chemical if the secretory cell and the target cell are nearby each other. They are not necessarily in contact, because remember, this is a form of indirect communication. But the secretory cell and the target cell are within one neighborhood. Therefore, the chemicals which are being released by the secretory cell travel via the extracellular fluid to the target cell. It's just through simple diffusion from the secretory cell. The chemical will just diffuse in the interstitial fluid to reach the target cell. If that happens, then we call that paracrine signaling, or that chemical will therefore be classified as a paracrine ligand. It's a ligand, but it's paracrine because being released by a cell within the neighborhood. You can also have what we call neurotransmitter. So what's a neurotransmitter? You call it a neurotransmitter if that chemical is being released by a neuron and it's going to another neuron. If the chemical being released is being released by a neuron, which means that the secretory cell is a neuron. If the secretory cell is a neuron and the target cell is a neuron, then you call that chemical a neurotransmitter. Like here, this is a neuron, that's another neuron, so that chemical, that ligand becomes a neurotransmitter. You can also have hormone. When do we call it hormone? A ligand is termed a hormone if that ligand is released into the bloodstream. Now, the criteria here is that the target cell is far away from the secretory cell. So the only way in which that chemical can reach the target cell is if that chemical is transported through the bloodstream. Therefore, the secretory cell must release this chemical into the bloodstream, and the bloodstream will then transport that chemical from the site that it has been released into. That means from the secretory cell, the bloodstream must transport this chemical all the way to the target cell. We can still revisit the concept I gave you about pituitary gland releasing growth hormone, which acts on some cells in the femur. There is no way growth hormone can travel all the way from the brain to your lower limb through diffusion. It will never reach. But this is made possible if that chemical is released into the bloodstream. So when chemical messengers are released into the bloodstream, we call those chemical messengers hormones. Such type of ligands are called hormones. Lastly, okay, there is another class that I didn't put there, but remember I'd mentioned neurohormones. So you call it a neurohormone when it is a neuron releasing the chemical into the bloodstream. Then you call it neurohormone. If a neuron is the one releasing that chemical into the bloodstream, you call it neurohormone. And I remember there was also another one called autocrine. You call it autocrine if the cell that has released the signal, the cell that has released the chemical, is also the target cell. That becomes autocrine. So don't confuse autocrine with paracrine. You call it autocrine if the secretory cell is also the target cell. So we may not want to call that indirect type of communication per se. It's a unique type of communication, but either way, it's a type of chemical signaling. Paracrine is where the target cells are within the neighborhood. Neurotransmitter is where a neuron releases a chemical, and that chemical goes to another neuron. Hormone is where a cell releases a chemical into the bloodstream to go to a distant target cell. Neurohormone is where a neuron releases a chemical into the bloodstream. You may have heard of pheromones. Those may not really apply in humans per se. Pheromones may not apply in humans per se, but they could apply in other animals. So let's not talk about them. Right, so the last thing I want to do is to give you examples of common chemical messengers as we finish. You can have chemical messengers which are amino acids. Usually there are 4 major amino acids which are used as chemical messengers in the body. And these 4 are examples of chemical messengers which are basically amino acids: glutamate, aspartate, glycine, and what we call GABA. GABA stands for gamma aminobutyric acid. You can just call it GABA. These are amino acids, which are chemical messengers. Gamma aminobutyric acid. There's some chemical messengers which are called amines. An amine is where you have two amino acids. All right. So you have histamine, serotonin, catecholamines. Catecholamines include things like adrenaline and noradrenaline. Then we have T3 and T4. These are thyroid hormones. T4 is what we call thyroxine. The name for T3 is very long. Don't care about it for now. You can just call it T3. It stands for triiodothyronine. I'm just giving you examples of chemical messengers, which are amines in nature. You can also have chemical messengers, which are proteins or glycoproteins. These are the majority, actually. So pituitary hormones like growth hormone, antidiuretic hormone, prolactin, those pituitary hormones, they are proteins or glycoproteins. Also, pancreatic hormones fall under this category. Insulin, glucagon, those are either proteins or glycoproteins. You can have steroids. Steroids have cholesterol within their backbone. So they have cholesterol within them. Examples of steroid chemicals are gonadohormones. So gonadohormones are hormones from the testes and ovary. That means they include things like testosterone. They include things like estrogen and progesterone. We also have corticosteroids. Corticosteroids are steroids from the adrenal cortex. Such include things like cortisol, it's a hormone. Aldosterone is a hormone. We can keep to those two for now. Lastly, we have what we call eicosanoids. Eicosanoids are chemicals which are formed from membrane lipids. The lipids in the cell membrane are the ones which form these chemicals. So examples include prostaglandins, leukotriene, and thromboxane. You'll come across these ones mainly even in immunology. They tend to be inflammatory markers. These ones, they tend to be inflammatory markers. So these are examples of common chemical messengers. The intention here is not to make you remember everything on this list. That is not the intention. The intention is to make you know that we don't just have one category of chemical messenger. There are many chemical messengers. So it doesn't really matter much if you don't remember examples of chemical messengers that I've given you here. I'll be happier if you know that chemical messengers can be many. And if you remember 2 or 3 examples, that's good enough. Right. So I'm done with the first part of the lecture. There's some questions I've projected for you there. I want you to discuss them, or rather not discuss, but just look at them and be sure that you are able to answer them. I'm just leaving them there for a minute. Then I go straight ahead to the next class. I don't think you're already tired. So I'll start the next session immediately before I'll give you a break at the end, in the middle. We have like 3 sessions today, so I'm done with the first one. I'll go direct to the second one, and then I'll give you a break between the second and the third. 1 minute, just look at that. If you have a question, also feel free to ask. Okay, so these quizzes are just for you, not for submission. Right, let's start the next topic. So the next topic is going to be body fluid compartments. This may not necessarily be part of cell biology, but you need to know body fluid compartments before we talk about the last lecture on cytology, which is on membrane physiology, largely talking about electrical activities on the cell membrane. For us to understand that topic that we are going to do next, you need to understand body fluid compartment. So body fluid compartment may not necessarily be part of cell biology, but we need to have it here before you understand the next lecture we're going to have. For those who feared mathematics, you might be in trouble now because you're going to do some mathematics kidogo. Now, this is what you're going to do. We are going to state how the total body water is distributed in various compartments of the body. That's one agenda. The other agenda is we are going to state the composition of the intracellular and extracellular fluid, the fluid within the cells and the fluid outside the cells. We are going to state their various compositions. So just 2 key agenda. Before then, let's define something. When we talk of total body water, what does it really mean? It means the total amount of water in the body. That sounds repetitive, but I think it's straightforward. The total amount of water in the body is what we are calling total body water. This total body water is found in various body fluid compartments. So there are multiple fluid compartments within the body that contains this water. the amount of the water that is in the body varies depending on other factors. Importantly, it depends on your age, it depends on your gender, it may also depend on your body weight. But I'll keep to these two, age and sex, as the key determinants of total body water. But I've alluded to you that even body weight affects total body water. How does it affect total body water? The age factor. Now, the age factor affects total body water this way, that the younger you are, the more the total body water. As you can see in this image on your right, a newborn has about 80% total body water. 80% of a newborn is just water. So if a mother delivers a child, anaanza kuringaringa ati mtoto ni mzuri sijui nini, you can just tell them 80% ni maji. All right, that's on a light note. So younger people will have more total body water. A newborn has 80% total body water. Older people will have less body water, but not really less. As a matter of fact, an adult male has 60% total body water. An adult female has 50% total body water. The reason why females have less water is because of fat content. Females will have more fat content than men, and that makes them to have less water because of the presence of fat. Usually they're a bit exclusive. Having said that, it also means therefore that if you're overweight with more fat, then you'll have less water. That is how weight also affects total body water. In as much as we've talked about different categories of people having different amounts of total body water, for the sake of medicine, for the sake of physiology, for the sake of pharmacology, where we need to calculate total body water to know how much drug someone should be given and the like, for the adult dosages of drugs, we usually make an assumption that the person who is being called an adult is someone who is an adult male. The calculation of drugs which are given are based on the assumption that everyone is male and everyone weighs 70 kgs. That is the physiological weight, 70 kgs. So I want you to understand that because it's difficult to take into consideration the different weights of people when you're manufacturing drugs, let's say augmentin, it is just adequate to say this is for adults. So what's our reference? The reference is usually an adult male who weighs 70 kgs. Sorry, my internet tipped a bit. I think it's back now. So I was telling you that for the sake of calculations of drug dosages are usually based on the assumption that the adult is an adult male who weighs 70 kgs, 70 kgs. But for the sake of this lecture, we are going to maybe talk about those differences, but we'll also be referring to the physiological man and the physiological weight quite a lot so that you familiarize yourself with that. So the key thing in this slide is for you to know that babies have more water than adults. Men have more water than female for the adults. Our reference is this one. And so whatever you're going to calculate today, we will be putting reference to the physiological one, which is 60%. That 60% of adults is made up of water. Now, I want to take you through those body fluid compartments. We've said that the total body water is within various compartments. The water within the body is either inside cells or outside the cells. So the terms we use for this are, it is either intracellular fluid or it is extracellular fluid. So the intracellular fluid is a fluid within the cells. Extracellular fluid is the fluid outside the cells. This image tries to explain that shortly. These are our cells. The fluid within here is intracellular fluid. The fluid here is extracellular fluid. The fluid here is also extracellular fluid because it is not within cells. This is within cells. This is outside the cells.
Please.
Sorry,
could you
Kevin?
repeat, please? We couldn't hear you.
Okay, which part do you want me to repeat?
Start from the intracellular fluid and extracellular fluid compartment.
Okay, so intracellular fluid is a fluid inside the cells. Extracellular fluid is the fluid outside the cells. So this one here is outside the cells, it is extracellular. The fluid here is also outside the cells, it is extracellular. We are going to give them names shortly. But you can see this fluid here is actually outside the cells, between cells. That is what is otherwise called interstitial fluid. The fluid here is outside the cells but within blood vessels. That is what is called plasma. There is another entity to the extracellular fluid that I'm also going to add shortly. But let's first talk about this intracellular fluid. So when we talk of intracellular fluid compartment, we are referring to the fluid within cells, as I've told you. This one, not this one. The fluid within cells is what we call the cytosol. Last time we had that lecture on cell biology. This fluid constitutes 2/3 of the total body water. Now I want to ask a question at that point then. We have said that the physiological weight of an adult male is 70 kgs. we have also said that the total body water for an adult male is 60% of the body weight. Having said that, I want you people to then calculate how much water is intracellular in someone who weighs 70 kg. Okay, do the calculation, then let me hear your responses. You can type there. So the responses you'll give me in liters. I'm waiting. I don't expect percentage. You give me in liters. Huh? So far, everyone who has written something is wrong. Okay, someone has gotten it. Make sure you get 28 liters. So the correct answer is 28. Now, I want to invite one of those people. Camilla, are you able to tell us how you got your 28?
Yes.
Okay, you can try.
So you said the person, the kg of person is 70 kg, and then the fluid in a man is 60%. So I multiplied 70 by 60 over 100. So I got 42 kg. Then intracellular fluid is 2 over 3. So 42 multiplied by 2 over 3, I got 28.
Thank you, Camilla. So that's how to do it. For those who didn't hear well, she said that the total body water is 60% of the weight of the person. So that means that if I weigh 70 kgs, then my total body water is 60% of 70 kgs, which means my total body water is 42 kgs. But you can just directly convert kgs into liters by multiply by 1, because 1 liter of water is 1 kg. You remember the density of water is 1 gram per whatever. So it means that the total body water of someone who weighs 70 kg is 42 liters. The total body water of someone who weighs 70 kg is 42 liters. So we're asking ourselves, if that is the total body water, 42 liters, then how much water is intracellular? So you take 2/3 of 42, and then you get 28 liters. That means that the intercellular fluid of someone who weighs 70 kg is 28 liters. Right, I want you to do the same, but now the weight of this individual is 90 kgs. The person weighs 90 kgs. How much fluid will be intercellular? You have a minute.
Male or female?
Now we agree that physiologically we're referring to the male. Eh? Okay, I think we are on the same track now. That's good. You realize that if the total body water is 2/3, sorry, if the intracellular fluid is 2/3 of the total body water, then it actually means that it is 40% of the total body weight. Because remember, total body water is 60% of total body weight. So 2/3 of that will be 40%. So you can still, you are 90, 40% of 90, you get 36. Even the other one, 40% of 70, you get 28. Great, that is intracellular fluid. I can see people answering. Okay, still the same thing, apart from those who have gotten 42. I think you've now known how we've gotten 36. Now let's talk about the extracellular fluid compartment. The fluid outside the cells. Now, extracellular fluid exists in 3 compartments. You have what we call the interstitial fluid compartment. That is one of the compartments of the extracellular fluid. What are we calling interstitial fluid compartment? The interstitial fluid compartment refers to the fluid that is immediately outside the cells. In our previous talk, this is what we called the internal environment. The interstitial fluid constitutes the internal environment. This fluid constitutes 75% of the extracellular fluid compartment. Right. I feel like giving you time to calculate that now. Let's say the person weighs 90 kgs. How much fluid will be interstitial in someone who weighs 90 kgs? So do that. Let me also do it so that I'm ready when you write something right or wrong.
We didn't get your question.
I want you to calculate the amount of interstitial fluid in someone who weighs 90 kgs. So That sounds hard for many people. I don't know why. Right, so the people have written 13.5 have gotten it. Nathaniel, do you want to tell us how you got your 13.5? I can see you're the first one who got it right there.
Okay, Mwalimu, this is how I got it.
Yes.
So when I picked 60% of that 90 kg person, I got 54 liters.
Mm-hmm.
So we've said that interstitial compartment consists of 75% extracellular fluid.
Yes.
So when you take 75% of that 54 liters of water, you get 13.5.
Really? 75% of 52 can't be 13.5.
Of 54, not 52.
All right. Yeah, I'm saying 75% of 54 cannot be 13. Hammer? You know 75% is 3/4.
Yeah, 75% is 3/4. Okay,
Yeah.
let me do something here.
Okay. Okay, someone else? How did you get your 13.5? 13.5 is the correct answer. But how do you get it?
Can I try?
Yes, yes. Carry on. Sorry, I didn't hear the person talking. Who wants to try?
Me.
Okay, try.
I took 60% out of 100 of 90 kgs.
Okay.
Then I got 54 liters.
Yes.
And when you said intracellular is 2/3.
Yes.
Therefore, a third is extracellular.
That's right.
So I said a third multiplied by 54. Then I got 18 liters.
Very good.
Extracellular. In the extracellular, you said it constitutes of 75%. Therefore, I took 75% of 18, which is 13.5.
Very good. Thank you, Kevin. So that's how to get it. We've agreed that total body water is 60% of the weight. So if you weigh 90 kg, then it means that 54 is 60% of 90. So 54 liters is the total body water. We've said that 2/3 of total body water is within the cells, intracellular, which means 1/3 is outside the cells, extracellular. So 1/3 of 54 is 18. If you weigh 90 kgs, the total amount of water that is outside your cells is 18 liters. Then now we are saying interstitial is 3/4 of the extracellular fluid. So you take 3/4 of 18, you get 13.5. Right. We didn't multiply with 75 twice. I think, okay, you want me to repeat that? I can repeat. We've said that what you need to do is just to first calculate the total body water in someone who weighs 90 kg. That's the first step you need to do. You get that by taking 60% of the total body weight. The next step is you need to calculate how much water is extracellular in this person. So you take 1/3 of their total body water. Then the last thing you do is to determine how much water is interstitial. So you take 3/4 of their extracellular fluid compartment. If you follow those 3 steps, you're going to get 13.5. The second fluid compartment, which is extracellular, is what we call the intravascular fluid compartment. Intravascular fluid compartment is a fluid within blood vessels. This fluid is also called the plasma compartment. It is a fluid within blood vessels, but don't consider the fluid within the blood cells, because the fluid within blood cells is still intracellular. Even though the fluid is within blood vessels, it is not within the blood cells. This fluid is 20% of the extracellular fluid volume. 20% means a fifth, 1/5. It's 20% of the extracellular fluid volume. I won't give you that to calculate. I think you now are familiar with how to calculate them. The last fluid compartment, which is extracellular, is what we call the transcellular fluid compartment.
Could you repeat intravascular?
Intravascular is within blood vessels. That is intravascular, the fluid inside blood vessels. All right. Transcellular fluid compartment is the fluid enclosed in chambers which are lined by epithelial membranes. There are some chambers which are lined by epithelial membranes. Those chambers are very many, but in total, transcellular fluid compartment constitute 5% of the extracellular fluid. Only 5%. 5% means 1 out of 20. Okay, let me repeat that. My internet tipped. So I'm saying transcellular fluid is a fluid that is enclosed within chambers lined by epithelial membranes. There are very many of such chambers in the body. Even though there are many, only 5% will actually be in these membranes, in these chambers, I mean. And I'm saying 5% is 1 out of 20. So 5% of the extracellular fluid is what we call transcellular. Now, when you talk of transcellular fluid, what do we really mean? We mean synovial fluid. This is the fluid within synovial joints. We mean intraocular fluid. This is the fluid within the eyes. It could be the aqueous humor. It could be the vitreous humor. We mean cerebrospinal fluid, CSF. This is the fluid within your central nervous system. It could be pleural fluid. This is the fluid within the pleural cavity, the one that lubricates the lungs. It could be pericardial fluid. This is the fluid around the heart. It lubricates the heart. It could be peritoneal fluid. This is the fluid in your abdomen around the intestines. Again, it lubricates the intestines from outside. And it could be digestive secretions, things like saliva, things like gastric acid, things like pancreatic juice, and the like. Now, I want you to look at this list, and you realize that I have not included urine here. Please don't include urine here, because urine is not a body fluid compartment. One unique thing about body fluid compartments is that these fluid compartments can actually redistribute from one chamber to another. If this fluid is within the blood vessels, it can actually go to interstitial fluid. Interstitial fluid can go back to blood vessels. It can go to the pleural fluid. Pleural fluid can go back to the interstitial and the like. But once fluid has reached urine level, it can't go back. That is why urine is not a body fluid compartment. So having said that, let me make a summary of what we've said about extracellular fluid compartment. We have said that there are three compartments in the extracellular fluid. One of them is what we've called the interstitial fluid compartment. We have said this is the fluid between cells. It constitutes what we call the internal environment. It makes up 75% of the extracellular fluid. We've mentioned another compartment being called intravascular compartment. We've said this is the fluid within blood vessels, so we call it plasma. And it contributes 20% of the extracellular fluid. And lastly, we've mentioned that there is what we call transcellular compartments. This transcellular compartment refers to fluid in epithelial line chambers. And I've given you examples of such chambers: synovial fluid, aqueous humor, vitreous humor, CSF, pleural fluid, pericardial fluid, peritoneal fluid, and digestive secretions. Even though there are many, in total, they constitute 5% of the extracellular fluid. You'll remember that when we were calculating extracellular fluid, we agreed that extracellular fluid is 1/3 of the total body water. Now that intracellular fluid is 2/3 of the total body water. So you can be given anything to calculate about total body water. Either this compartment, this compartment, this compartment. Most of the time, there'll be those ones, because intracellular is straightforward. Then you are given the weight. So what you need to know is to understand that our reference is 60% of total body weight. For you to know the total body water, you use 60% of the total body weight. Right. Okay, I think that was remaining, but I've already told you that the transcellular fluid is intraocular, which means aqueous humor and vitreous humor. Synovial fluid. Mesothelial fluid is fluid in those pleural cavity, peritoneal cavity, and pericardial cavity. That's what we are calling mesothelial fluid. Pleural cavity, peritoneal cavity, and pericardial cavity. Let's do something last before now I give you a break. I want us to look at the composition of the body fluids. And I want us to focus largely on the intracellular fluid and extracellular fluid. But before that, on this graph, which I don't want you to pay much attention, is just for you to understand that the interstitial fluid and plasma, remember those are part of extracellular fluid, They are very near each other. That's what I want to get from here. They are very near. Almost similar. Almost similar. Almost similar. Almost similar. The only difference is their protein contents. That's the only difference between interstitial and plasma. The protein content is the one that is...
Could you repeat, please? I think your internet connection was a bit lost.
Okay, I'll repeat. So I'm saying, what I want you to get from this slide is that interstitial fluid and plasma are almost similar in terms of their contents, in terms of their composition. For most of the things, if you look at sodium, they're almost similar. Chloride, they're almost similar. Bicarbonate, potassium, phosphate, calcium, magnesium. The only difference is on their protein composition. That is where now we have more proteins in the plasma compared to the proteins we have in the interstitial fluid. This is the only place where there's significant difference between intravascular and interstitial. Now that is what I wanted to pick from this slide. But now I want us to now pick the differences between the intracellular and the extracellular fluid in general. I will use the next slide to show that difference. So we are looking at two fluid compartments here, the extracellular fluid and the intracellular fluid. We are also looking at positively charged ions and negatively charged ions. So there are some questions I want you to tell me from the graph you're seeing here. What is the main intracellular cation? The main intracellular cation is which one? Anyone? The main intracellular cation is which one?
Can I try?
Yes.
Potassium ion.
Yes, potassium.
Potassium.
So potassium is the main intracellular cation. I want you to get that right. And therefore realize that if potassium was to diffuse, remember from our lecture on leaky channels, if potassium was to diffuse through its leaky channels, it will then tend to move from inside going out of the cell. Because potassium is the main intracellular cation. I also want you to note that the main extracellular cation is sodium. That means that if sodium was to move along its concentration gradient, constantly sodium will be trying to move into the cell from outside through diffusion. I want you to pick those two. You can ignore these ones. But the other one I want to pick is this one. Chloride is the main extracellular anion. That means that if chloride was to move, it will tend to move into the cell many times because it's more abundant outside. There are two anions which are inside the cell that I want you to pick. Phosphates and proteins. There are more proteins and phosphates inside the cell than outside the cell. And remember, they are negatively charged. They are negatively charged. They are more inside the cell than outside the cell. I'm insisting on those ones because these are the ones we want to know in our next lecture. We want to know that proteins and phosphates are more inside the cell than outside the cell because there will be implications of that concept. So on this slide, I want you to have a take-home message that the major intracellular cation is potassium. The main extracellular cation is sodium. The main extracellular anion is chloride. The major intracellular anions are phosphates and proteins. They are negatively charged ions. Good. When we take laboratory measurements of these chemicals, most of the time the laboratory estimates that we get will be the estimates of the plasma. So when you send someone for laboratory test, this is what will be measured. This one, the plasma one, because the samples you take are blood samples. You don't take interstitial fluid sample. You don't take intracellular fluid sample. You take plasma sample. So in that, even though you see here that potassium is so high, but it's high inside the cell. Otherwise, if you take blood sample of potassium, it will give you this one. Potassium is usually very low. Extracellular. Actually, if you want to kill someone, just give them potassium. They'll not even last 20 seconds. You'll be taught that. I'm not teaching you to kill. You'll be taught that. All right. So I want to finish this, but let me give you an assignment first. Again, you're not submitting this to me. You will, at your own time, state the key functions of the major anions and cations in the body. And just focus on the previous slide for you to know which anions, which cations. So this slide. So look at what's the role of sodium, how about potassium, calcium, magnesium, chloride. You can forget about phosphate. Okay, you can forget about these three. Let me repeat that. So I'm saying, just look at the major, this one's the first one up to here. What you're going to do is to state their role in the body. You're also going to state their actual concentrations, and the concentrations should now be the laboratory concentrations, which means they're concentrations in plasma, or basically they're extracellular concentrations. So that means when you talk about potassium, this is not the value you're going to give me, because this is intracellular, but this is the value you're going to give me. How much is potassium within the plasma? Because this is the reference ranges that now you need to be aware of so that you're able to interpret laboratory results of patients. You will look at the concentrations of the different ions in plasma. Then you'll set the functions of those different ions in the body. that would be your assignment. I want to give you a break now. When we come back, we are going to look at membrane potentials. But before that, I hope you people are able to answer those questions that I've projected. Okay, I think they're straightforward. We've already done this one, I think. No, we didn't do it, but I know you can do it on your own. This one I also believe you can do it on your own. Straightforward. So, is there a question before I give the... Okay, no question. So let's take how many minutes break? It's now 9:32. So let's come back at 9:45. 9:45, we are going to start on membrane potentials. You can take your break. We can resume now. We are now going to look at membrane potentials. This lecture requires a lot of your attention, so try to follow. If I lose you, just stop me and I'll repeat. What you're going to learn here, we are going to define what are membrane potentials. We are also going to look at the varieties of membrane potentials. We are going to state the ionic basis for what we call resting membrane potential. Then we are also going to explain how action potentials are generated. And lastly, we are going to outline the properties of action potentials. Action potentials are the ones we are calling propagated potentials. So this is our agenda this morning. Let's begin by defining what membrane potentials are. Membrane potentials just refers to electrical charges that exist across the cell membrane. Electrical charges that exist across the cell membrane is what we are calling membrane potentials. So that means that there are some charges that exist across the cell membrane. The cell membrane is not neutral in terms of electrical charges. These charges are usually measured in millivolts. So the unit of measurement of the electrical charges across the cell membrane is millivoltage. How do these charges come about? These charges come about because of the asymmetric distribution of charged particles across the cell membrane. We have just seen in the previous session that potassium is a positively charged ion and it is more inside the cell compared to outside. We have also seen that sodium is a positively charged ion, but it is more outside the cell than inside the cell. We've noted also that chloride is a negatively charged ion, and it is more outside the cell than inside. Likewise, we've mentioned something about the proteins, which are negatively charged ions, and they are more inside than outside. So if you look at those ions I've talked about, they are charged either positively or negatively, but they are not symmetrically distributed across the cell membrane. They are those which are more on one side than another. So it's because of this asymmetric distribution of charged particles across the cell membrane, it will then lead to the membrane having some charges across it. Membrane potentials are created by this asymmetric distribution of charged particles. There are three types of cell membrane potentials. We have what we call the resting membrane potential. We have what we call the graded potentials, and we have what we call action potentials. Action potentials are also known as propagated potentials. So I want us to then look at these three types of membrane potentials in the order in which I've listed them. That means we can begin with resting membrane potential. If I was to define for you the resting membrane potential, I will say it is the overall charge that exists across the cell membrane when the cell membrane is not disturbed, when the cell membrane is at rest. It is the overall charge that exists across the cell membrane when the cell membrane is not disturbed. The type of disturbance I'm talking about here is electrical disturbance. So there's nothing that is disturbing its equilibrium. It is the equilibrium that we achieve, that cell membranes achieve. As I've mentioned, it will be measured in millivolts. So that overall charge is measured in millivolts. This is the cell membrane. So we see charges here. Maybe you're wondering, why am I putting positive here and negative here? We are going to see that shortly. But what I want you to pick at this point is that there are some charges either way across the membrane. And that can be measured in millivolts. So as long as this cell membrane is not disturbed, the charges that exist across it will be termed the resting membrane potential. I want us to now look at how the resting membrane potential is generated. How is the resting membrane potential generated? One concept that I want you to build from now henceforth is that the cell membrane is permeable to potassium, to sodium, and to chloride. This permeability is through their leaky channels. We said leaky channels are channels which are always open. If leaky channels are always open, then it means that ions can freely move through these leaky channels from regions of higher concentration to regions of lower concentration. Having said so, remember that potassium is more inside the cell, sodium is more outside the cell, chloride is more outside the cell, but they are free to move through their leaky channels to the other compartments where the concentration is less. The images here show you those leaky channels. The leaky channels are usually specific, as we agreed last time. So you'll have potassium leaky channels, sodium leaky channels, and chloride leaky channels. Now let's see how these molecules then will be leaking. Let's consider this to be the cell membrane, and this side to be the extracellular fluid, this side to be the intracellular fluid. Let's also consider that this is the potassium leaky channel, this is sodium leaky channel, and this is chloride leaky channel. I want to see how potassium will leak through here. And especially, I want to ask myself the direction of that leakage. And this I want one of you to tell me. So if potassium was to leak, in which direction will potassium leak? Anyone?
Towards the extracellular fluid.
Thank you, Linda. Potassium will move outwards. The reason why potassium moves outwards is because potassium is more inside the cell than outside the cell. Right. How about sodium? How will sodium move? Anyone?
Sodium will move inside
towards intercellular fluid.
Very good. So sodium will move inwards, and that will also be similar to chloride. The term we use for a molecule that is moving outwards is efflux. So we will have potassium efflux. Efflux is the term we use when there is net movement outwards. The term we use for net movement inwards is influx. So for sodium and chloride, we constantly have influx. For potassium, we constantly have efflux. Remember, these are leaky channels, which means they're always open, which means potassium is constantly moving out of the cell. It also means that sodium is constantly moving into the cell. Chloride is also constantly moving into the cell. I'm expecting someone to then start asking themselves, if potassium is always moving out of the cell, at what point will it stop? Or will we deplete the intercellular fluid? So I've already asked that question for you. At the end of this lecture, you should have an answer to that question. Now, having agreed that those ions move in the directions that we have seen, we want to see the impact of those movements in terms of the charges on the cell membrane. Today you'll forgive my neighbor's dog. We want to see the impact of the efflux of potassium, the influx of sodium and chloride on the charges on the cell membrane. I hope you remember also that potassium is a cation, sodium is a cation, chloride is an anion. Now, we have said that generally potassium is more inside the cell than out of the cell. This blue region represents inside the cell. Then this is extracellular, this is extracellular. We have said that potassium will tend to move out of the cell. Being a positively charged ion, if potassium moves out of the cell, which means if we have potassium efflux, what does that mean in terms of the membrane charges? It means that potassium will be carrying positively charged ions out of the cell. I want you to grasp that. That as potassium moves outwards, it will carry with it positive charges out. If it carries positive charges out, it means that the inside of the cell will be more negative if you compare that with out of the cell. So what is the effect of potassium efflux? The effect of potassium efflux is that the inside of the cell becomes more negative, or we can say the outside of the cell becomes more positive. Anything that causes that kind of charge asymmetry is termed as being polarizing. Polarization is the making of the inside of the cell be more negative and the outside be more positive. So any ion that makes the inside of the cell be more negative is termed as causing polarization. Membrane polarization is when the inside becomes negative compared to outside. I hope you've understood that. That is membrane polarization. So potassium efflux causes membrane polarization. And I've defined polarization as when the inside becomes more negative, the outside becomes more positive. Right. Potassium efflux causes membrane polarization. Let's see what chloride does. And this one I want one of you to try. So the question is, chloride is a negatively charged ion. Chloride is more outside the cell. My question to you is, if chloride was to move through its leaky channels, will chloride movement cause polarization or depolarization? Anyone? And maybe the reason is more important than the answer. Anyone just try?
Maybe try.
Yes, Linda.
The chloride ions will cause depolarization too,
because they also carry negative charges, making the inside of the cell to be more negative.
Okay, so you said chloride movement will cause polarization or depolarization? Which one?
Polarization.
Polarization. Thank you, Wamboi. So we are saying that chloride is negatively charged and it's more outside. That means if chloride leaks through its channels, it will be an influx. It will be carrying negative charges into the cell. And that means it will be making the inside be more negative and the outside be more positive. Therefore, chloride influx also causes membrane polarization. Now let's look at sodium. I want someone else to try. If sodium moves through its channels, what will sodium movement cause? Is it polarization or depolarization? Yes?
Depolarization.
It will cause
It will cause
depolarization.
depolarization.
All right, I agree with you. So, sodium, because sodium is positively charged and is more outside, if it moves, you are going to have sodium influx. That means the inside of the cell will be more positive. The outside of the cell will be more negative. So sodium causes the inside of the cell to be more positive and the outside of the cell to be more negative. Because sodium is positively charged, it's taking positive charges into the cell. That means that sodium does not polarize. Sodium influx does not polarize. As a matter of fact, sodium influx causes depolarization. So depolarization is when we make the inside be more positive, or if you make the outside be more negative. It's like you are canceling the polarization. So at that point, therefore, we can argue. We have things which cause polarization. We have things which cause depolarization. We can argue that then they'll cancel each other. But that's not true. The reason why that's not true is because in as much as we have factors that polarize and factors that depolarize, these ions do not diffuse across the cell membrane at the same rate. They do not diffuse at the same rate. As a matter of fact, when the membrane is at rest, potassium efflux is several times faster than sodium influx. Actually, about 50 times faster. That means that for every sodium that enters the cell, 50 potassium will have moved out of the cell. For every sodium that enters the cell, for every single sodium influx, there is 50 potassium efflux. Now, with that in mind, I want you to tell me, between polarization and depolarization, which one will override?
Polarization.
Polarization will override. I agree. Because in as much as sodium depolarizes, it is outweighed by the effect of potassium. So polarization overrides. Now, back to that important question I asked you. Your potassium is always moving out of the cell. There is a risk of depleting potassium inside the cell then. However, potassium is never depleted. Also, sodium is never depleted inside the cells. Sorry, outside the cell. It's never depleted. I want someone to tell me why. Okay, that could be a very difficult question, but maybe someone has already read. Why do you think potassium is never depleted inside the cells and sodium is never depleted out of the cells? Anyone? Just give a trial. It's something you can't.
Maybe I try.
Yes, yes.
Because you've said the efflux and efflux take place through the diffusion and in the leaky channels, and the diffusion is based on concentration. So when the concentration balance in that the outside and the inside have something like equilibrium, the diffusion will stop. There will be no more diffusion since there is a balance, and the concentration will be the same.
Thank you very much, Wamboy. You've really done well. So that's one of the reasons why you can never deplete. The fact that these are diffusions, so you'll definitely reach an equilibrium. Now, the other thing I want to tell you is that the equilibrium we are going to achieve here is not just based on the concentration of the specific ions. It is also based on the charges. When you have a lot of positive charges out, in as much as the concentration of potassium will still be higher here, and so it wants to move out, those positive charges will actually repel the positive potassium ions that are moving. So you'll actually achieve equilibrium of concentration, or rather, let me say it this way. The concept of equilibrium of concentration, as well as the concept of equilibrium of charges, will not really allow us to deplete potassium inside the cell. But then that makes us ask, so does it mean that it will reach a time that there's no movement at all? And maybe the answer is no. There's always movement. So why is it that there's always movement? That brings me to the second reason why we never deplete potassium and sodium in their respective compartments. Now, the reason is this. There is something we call the sodium-potassium pump. Sodium-potassium pump is something that pumps sodium and potassium against their concentration gradients. That means that it's a mechanism of active transport. It's a mechanism of active transport. How does it actively transport sodium and potassium? We know that sodium is more out of the cell. So if you're going to have active transport of sodium, it means that this pump will be moving sodium out of the cell. We also know that potassium is more inside the cell. So if you're going to have active transport of potassium, it means that this pump is going to cause potassium influx, not efflux, like what the leaky channels do, but influx. Now, this is what the pump does. In as much as it pumps those two ions, again, it does not pump them at the same rate. It will pump 3 sodium out for every 2 potassium in. Now, I want you people to look at that pump again with what I've told you about how it pumps. And tell me, what should be the effect on the charges, the net effect on the charges, if I take 2 potassium in for every 3 sodium out? Remember, sodium and potassium are both positively charged ions. Anyone?
Okay, I think the cell membrane will polarize.
It will cause polarization. I agree with you. So Nathaniel, why would the cell be polarized? What makes you say that sodium potassium pump polarizes the membrane?
Okay, this is because both the inside and the outside is positive.
Okay, that is not the definition of polarization though, Nathaniel.
Maybe I say, since you're pumping out 3 sodium ions, which means there are more positive ions being pumped out. So the outside of the cell will have more of the positive ions, causing polarization.
Very good. Thank you, Omboy. So that's the point, that this pump is taking 3 positives out for every 2 positives in. That means that the net movement is 1 positive out. It is causing positive charges net to be out, which means that there's net negative charges in. So the sodium potassium pump, because of the nature in which it is transporting the ions in question, it will also have a polarizing effect on the membrane. Good. I want you to remember something else. Okay, before we tell you what we remember, so we've said 3 things here so far: that potassium efflux causes polarization, chloride influx causes polarization, sodium potassium pump also causes polarization. Those are the 3 things we've said which cause polarization. In as much as sodium causes depolarization, the effect of sodium is outweighed by potassium. So we can ignore that for now. So because of these things, so far 3 things that cause polarization, we can say that the cell membrane is actually polarized at rest. One addition to add is that we mentioned that we have a lot of anions within the cell compared to out of the cell. And the anions we talked about are phosphates and proteins. Because we have a lot of negatively charged ions inside the cell, these ones also contribute to the inside of the cell being negative, which means they also contribute to the membrane polarization. Therefore, at rest, the cell membrane will be charged. with inside being negative and outside being positive. If that is measured, it will give us the resting membrane potential. So, if I was to summarize for you, 4 key factors that contribute to the resting membrane polarity, we will say one of the factors is this, that potassium efflux is there and is several times faster than sodium influx. So the effect of sodium influx is not felt really. The second factor is that we have sodium-potassium pump that pumps 3 sodium out for every 2 potassium in, so the effect is that we have polarization. The third factor is chloride influx, negative charges entering. And the fourth factor is that we have more anions, negatively charged particles, within the cell than outside the cell. That means that at rest, the membrane is polarized. The membrane is charged at rest, and the charge is in such a way that the inside is negative, the outside is positive. Great. If you are to measure the resting membrane potential, we agreed it's measured in millivolts, but this measurement varies depending on the tissue. I will teach you 3 of them because this will form the basis of our next series of lectures in physiology. The resting membrane potential in neurons is negative 70 millivolts. The resting membrane potential in cardiac muscle is negative 85 millivolts. And the resting membrane potential in skeletal muscles is negative 90 millivolts. For the other cells, it varies, but not even important to know them because we'll not be talking about their physiology much. But these 3 will be talking of their physiology, and that resting membrane potential will be important to take into account. So remember these 3 measurements. Maybe the other thing I forgot to tell you is that all cells, as long as they're alive, they will have a resting membrane potential. But the measurements I've given you are just for these 3 types of cells. Right. We are done with resting membrane potentials. Let me teach you about graded potentials before I give you some short break to think about something as I take the attendance. So, graded potentials, what are they? Graded potentials refer to transient disturbances on the normal resting membrane potential of a cell. You have a transient disturbance, which means a short-lived disturbance of the RMP. This disturbance only involves a small region of the cell membrane. The point here is it does not affect the whole cell membrane of that cell. It just disturbs a focus, a small region of that cell membrane. And that's the key thing. It is not diffuse. It is localized. It's focal. This type of disturbance can be either a depolarization disturbance, which means that if the resting membrane potential was negative 70, maybe it's now negative 60. You know, that's a depolarization because you are canceling the charges. It can also be a hyperpolarization, which means if it was at negative 70, maybe it's now negative 90. It depends on where we're starting from. So for example, if I give examples of neurons, neurons are at negative 70. if you have a stimulus that makes the cell membrane polarity to go towards -50, it means it is trying to depolarize because it's trying to make it go towards 0. That's depolarization. But if the resting membrane potential was at -70, then you have a stimulus that makes it even be more negative. We use the term hyperpolarization to mean it's making it more polarized. Hyperpolarization. So graded potentials can either be a depolarization effect or a hyperpolarization effect. Point is, it's a focal disturbance of the membrane. It does not affect the whole of the cell membrane. So the other parts of the cell membrane will just retain their normal resting membrane potential. If it's a neuron, it will just retain at 70. But at that point where there was a stimulus, you can have brief disturbance. So the amplitude of the disturbance will therefore depend, or rather, the amplitude of the disturbance will depend on the size of the stimulus. For it to move from -70 to wherever it's going, what will determine whether it reaches -80 or -90 is the size of the stimulus. So a weak stimulus will disturb it briefly, kidogo. But a stronger stimulus will have a higher amplitude of disturbance. That should make sense. If the stimulus is very weak, you'll just have a small disturbance. If the stimulus is very strong, you'll have a bigger disturbance. Great. I want to now talk about action potentials, but I want to give you a question first that you're going to do in your groups as I collect attendance. So the question I want to give you is to discuss the differences between graded potentials and resting membrane potentials. State the differences between graded potentials and resting membrane potentials. I'm dividing you into those groups right now. So there's something that will pop out in your screen. Just accept and be in one group. So I'm dividing you. You are 60 at the moment. I'm dividing you into 10 groups. You'll be 6 each. Something will pop in your screen. Just accept and be in your group. Right. I hope you've answered that question now. So in the next 20 to 30 minutes, we are going to talk about action potentials. And that will be the last part of the lecture. What are action potentials? Action potentials are rapidly propagated depolarization disturbances of the cell membrane. Get that right? It's rapid, which means it's happening very fast. They are propagated, which means they are moving along the cell membrane. It's not a constant thing. It's a wave that is moving. It's a depolarization, which means it is a time when the inside is positive, the outside is negative. That is a depolarization. Remember, polarization is when the inside is negative, the outside is positive. So, for those who may not have difficulty with the video, you can see in this neuron, there's a resting membrane potential where the inside is negative and the outside is positive. But there's a brief disturbance, if you focus on at the level of the arrow, where you have positive charges inside and negative charges outside, but a brief one. That is a region of depolarization. But we also appreciate in this video that that depolarization is being propagated. In this one, it's moving from this region, the cell body of the neuron, along the axon to the end of the axon. It's a depolarization disturbance, and it is propagated rapidly from one end to another. Those are action potentials. What causes action potentials? Action potentials are caused by sodium influx. The other ion that can also cause action potential is calcium influx, but let's not talk about calcium for now. For now, let's just focus on sodium. Sodium influx is the one that causes depolarization, as we've seen when we were discussing the resting membrane potential. But that means that you must have a lot of sodium that overrides the effects of potassium efflux for it to cause a depolarization. And we are going to see that that actually happens. Now, that disturbance of depolarization usually spread along the cell membrane. So it is not like graded potential that is just on a particular focal point. This one spreads along the cell membrane. It's diffuse, so to speak. In as much as resting membrane potential and graded potentials are experienced by all living cells, action potentials are only experienced by muscles and neurons. Only muscles and neurons experience action potentials. Now, tissues which experience action potentials are termed excitable tissues. So action potentials are a reserve for excitable tissues, tissues which can generate action potentials are just muscles and neurons, nothing else. What is the importance of action potentials? Action potentials are important for transmission of signals. That means that we'll have signal transmission in neurons. We'll also have signal transmission in muscles. Those are the ones that can transmit signals along the cell membrane. These signals are electrical signals along the cell membrane. Now, I want to see how action potentials are generated. Let's look at this. Consider this to be inside the cell, and this is outside the cell. The membrane at rest is positively charged out and negatively charged in. So this is the resting membrane potential. It's the resting state, normal for neurons. It's at -70. For muscles, it's at -85 for cardiac or -90 for skeletal muscle. Point is, this is the resting membrane potential. Now, we have sodium, and we know that sodium is more outside the cell than inside the cell. We also know that we have what we call voltage-gated sodium channels. Now, I'm bringing a new concept. When we talked about transport across the cell membrane, we mentioned that we have leaky channels, gated channels, and pumps. We've talked about leaky channels. Sodium leaky channel, potassium leaky channel, chloride leaky channels. We talked about them. Now I'm bringing the concept of gated channels. For gated channels, we said that you can have voltage gated, ligand gated, and mechanically gated channels. I'm bringing the concept of voltage gated sodium channels. We have voltage gated sodium channels, usually on the cell membrane, and that means that they're always closed, but they can be opened if there's a stimulus. Now, if we have a graded potential, that this causes disturbance on the resting membrane potential, that means that that graded potential is causing some voltage disturbance of the cell membrane. If that disturbance reaches threshold for a particular voltage-gated channels, then it can actually open a voltage-gated channel. Now, in this case, if you have a graded potential that causes some voltage disturbance, it may cause the sodium voltage-gated channels to open. Now, if you have opening of voltage-gated channels, what will happen? You are going to have a massive sodium influx. If you open voltage-gated sodium channels, you're going to have massive sodium influx. This massive sodium influx is unmatched. It cannot be cancelled by the regular leaky channels of potassium. Remember, for generation of RMP, we said that the leaky channels of potassium outmatch the leaky channels of sodium, and so polarization overrides. However, if you open voltage-gated sodium channels, depolarization will override. because these channels are very massive, very big, and you've opened them. So what will happen is that you're going to have sodium influx, massive, and that means that at that level you're going to have depolarization. What is important for you to note also is that as long as you've depolarized this region of the membrane, it means that the voltage gated channels here are also going to be opened. And that means you're going to have sodium influx somewhere there. And that will make the voltage gated channels here also to open. And you're going to have sodium influx again. Now, I'm bringing the concept of propagation, that as long as you've opened voltage gated channels in a particular region, it will affect the neighboring membranes, and so it will affect the neighboring voltage gated sodium channels, and that means they're going to open, and so the depolarization is going to be propagated. Usually, during the depolarization, there's even a brief reversal of the membrane polarity. What I mean is that the inside become positive, the outside become negative. Remember, for normal state, the inside is negative, the outside is positive. At the peak of an action potential, the inside becomes positive, the outside becomes negative. You've reversed the polarity. It is important for you to note that the depolarization sequence spreads along the cell membrane to the end because of, as long as you have a disturbance in this segment, you're going to open, yes, this one has already been opened, but you're also going to open voltage gated channels here because of the same effect, that there's a voltage disturbance that makes the voltage gated channels to open. And so when they open, they affect a membrane on a wider range, then that means that more voltage gated channels are opened again, and so the depolarization spreads throughout. That is why action potentials are propagated potentials. They spread throughout the membrane to the end. This is the concept of signal transmission. However, when the membrane is depolarized, it's a very unstable state for the cell membrane. Very unstable state for the cell membrane. See, the membrane is depolarized here. The depolarization is spreading less in that direction because this one is still the resting membrane potential. This is a very unstable state for the cell membrane. Very unstable state for the cell membrane. So what happens? When the membrane is depolarized, it will cause voltage-gated potassium channels to also open. Now tell me what will happen if you open voltage-gated potassium channels. It means that you are going to have massive potassium efflux. This massive potassium efflux is going to try to correct the membrane disturbance by making the inside be negative and the outside be positive. So basically, when you have this massive potassium efflux, you are trying to restore the resting state of the cell membrane by making the outside be positive and the inside be negative. So yes, potassium efflux will still come by opening voltage-gated channels so that the resting state is restored. But either way, the sodium influx came faster, and so depolarization occurred, and it's even spreading along the cell membrane to the end. So the signal will be transmitted either way. Then now the cell membrane needs to go back to its resting state. This is the whole concept of action potentials and how they are propagated. Remember, the first one must be sodium influx that causes depolarization. Then later on, potassium efflux comes to restore the membrane to its normal state. Let's look at the phases of an action potential. And I want us to understand this graph. We can, if you are to draw a graph of the events that occur during an action potential, we will have such a graph. So the example given here is that of a neuron, because you can see the resting state is at negative 70 millivolts. So let's take that to be the resting state, negative 70 millivolts. This region here represents the resting membrane potential. We have given the ionic explanation that is behind the resting membrane potential. We have said that the resting membrane potential is caused by 4 factors. Potassium efflux several times faster than sodium influx through their leaky channels. We've also talked about the effect of sodium-potassium pump. The third factor is chloride influx. And the fourth factor is that you have more negatively charged anions inside the cell. So that maintains the resting membrane potential at that level. If you have a graded potential that disturbs the membrane polarity, and that disturbance reaches a particular threshold. In this case, from this diagram, the threshold seems to be 15 millivolts. The difference between 70 and 55 is 15 millivolts. Usually the threshold can be anything between 10 to 20. So 15 is a good average. If the graded potential has an amplitude that enables the membrane to be disturbed by about 15 millivolts difference, then that disturbance can cause opening of voltage-gated sodium channels. If you have opening of voltage-gated sodium channels, you are going to have massive sodium influx. And that means that the membrane polarity, you are going to have a depolarization. So it's going to reach 0 and even make the inside be positive compared to the outside. So this phase of the action potential is what we call the depolarization phase. You can also call it upstroke. The stroke is going up, upstroke, or the depolarization phase. The depolarization phase of the action potential is due to massive sodium influx following opening of voltage-gated sodium channels. Now, when the voltage-gated sodium channels open, sodium ions will move in massively, and that will cause the membrane polarity to be depolarized. It will cause a depolarization. When the membrane is depolarized until the inside becomes positive, that has an effect on 2 things. One of those is that it will actually make the voltage-gated sodium channels too close. the moment the membrane polarity is reversed, voltage-gated sodium channels close. If voltage-gated sodium channels close, it means that we cannot continue having the upstroke. So we'll stop at some point. That peak of the action potential, where the pointer is right now, is what we call the overshoot. So what causes overshoot? Overshoot is caused by closure of voltage-gated sodium channels. Voltage-gated sodium channels close to cause that region there overshoot. At the same time, when you have the membrane in a depolarized state, it's a very uncomfortable state for the cell membrane. And so one of the effects it has is that it will make the voltage-gated potassium channels to open. If you have opening of voltage-gated potassium channels, you're going to have massive potassium efflux. Massive potassium efflux will make the cell membrane to now repolarize again. It will cause polarization. So we are using the term repolarization because it was polarized, then depolarized, so we are now repolarizing. And so this phase of the action potential is known as the repolarization phase. Repolarization phase is caused by massive potassium efflux following opening of voltage-gated potassium channels. Something interesting, when you open voltage-gated potassium channels, they remain open even beyond -70. The channels remain open. Usually, they will close at around -80. I want you to get that right, because I will ask you a question with regard to the pattern we see in neurons, the pattern we see in skeletal muscles, and the pattern we see in cardiac muscle, based on what I've told you. Voltage-gated potassium channels usually remain open up to -80. When it reaches -80, that's when they close. So in neurons, where the resting membrane potential is -70, when repolarization reaches -70, the voltage-gated potassium channels are still open. And because they're still open, it means that you are still going to have polarization. Now, when you're polarizing beyond the resting state, then we use the term hyperpolarization, or other is called the trough. So this phase of the action potential is called hyperpolarization, and it's caused by the delayed opening of voltage-gated potassium channels. Delayed opening. They open in a delayed manner. They are still open. These are the phases of the action potential. Of course, if the membrane polarity reaches -80, the potassium channels, the voltage-gated potassium channels will close, and then that means that the factors that explain the resting membrane potential will come to be, and so the resting membrane potential will be restored. These are the phases of an action potential. So what I want you to take note is that in as much as we have hyperpolarization in neurons, we will not expect to have this in cardiac muscle, because the resting membrane potential for cardiac muscle is minus 85, somewhere here. So this potassium, voltage-gated potassium channels will close way before the resting membrane potential is established. And that also applies to skeletal muscles. Now I want us to finish by talking about properties of an action potential. One of the properties of an action potential is that an action potential can only be triggered by a depolarization. If you have a graded potential, we agree that graded potentials can either be a depolarization or a hyperpolarization. Hyperpolarizations cannot generate an action potential because, look at this. If this is the resting state, if you have a graded potential that is causing the membrane potential to go in this direction, hyperpolarization, that cannot cause sodium channels to open. It will not cause voltage-gated sodium channels to open. Voltage-gated sodium channels open only at around that level in this case. So if the disturbance is towards the direction, it will not make the sodium channels to open. And that is why we are saying it can only be triggered by a depolarization, not a hyperpolarization. The second property of action potentials is that action potentials have a threshold that will initiate them. Basically, what this means that there's a threshold voltage that can make voltage-gated sodium channels to open. That threshold voltage is between 10 to 20 millivolts. So if the resting potential was at 70, it means that if you disturb that resting potential to make it 62, it means you'll have disturbed by about 8. That cannot generate an action potential in as much as it is a depolarization. That disturbance must be between 10 to 20. So that means if the resting potential was at negative 70 and you make it be now negative 50 something, so the threshold here is within this range, that will cause an action potential. But if the threshold is kidogo like A and B, it doesn't reach the threshold. If the stimulus is low, it doesn't reach the threshold, this will not cause action potential. A and B cannot cause action potential because they are subthreshold. C and D are above threshold, so they can cause action potentials. The third property is that action potentials are all or none events. What that means is that you either have an action potential in totality or it doesn't occur at all. You cannot have a weak action potential. You cannot have a stronger action potential. It's an all or none event. It's either it's present or it's not present. So, like here, either there is no action potential or there is action potential. And if it's there, this will not be now bigger than this one because now this one was a bigger stimulus. No. It's either present or absent. Action potentials also have a constant amplitude. Now, this is the amplitude of an action potential. That amplitude is usually about 100 millivolts. Action potentials have a constant amplitude of about 100 millivolts, which means what? If the resting potential was at negative 70 here, we expect the overshoot to reach positive 30, so that the amplitude is 100. If the resting potential was minus 90 here, we expect the amplitude, the other side, to be at positive 10, so that we have an amplitude of 100. They have a constant amplitude, irrespective of the amount of the stimulus. So this is different from graded potentials. The other property is that during an action potential, there is reversal of membrane polarity. I told you that. This is at -70. There is depolarization with a reversal of membrane polarity. So here, the inside becomes positive and the outside becomes negative. You've reversed the membrane polarity transiently. The last one is that action potentials have a refractory period. Now let's look at this graph as I teach you refractory period. The refractory period is a period within which it is difficult to initiate another action potential. What that means is that when an action potential is occurring, and let's see from here, an action potential is occurring, it is difficult to initiate another action potential on top of this one. You cannot superimpose action potentials. So during this period where this action potential is taking place, the membrane is refractory to the generation of another action potential. Now, refractory periods are of two types. We have the absolute refractory period, where it is not possible completely to generate another action potential. So that applies for depolarization phase, overshoot, and repolarization phase. During those three phases, the cell membrane is completely refractory, which means you cannot generate another action potential. However, beyond this, during the undershoot, during the hyperpolarization phase, you can generate an action potential, but you have to use a stronger stimulus. And the concept is this. When the membrane is at hyperpolarization, it will take a lot of energy for this membrane to be disturbed up to this threshold. See, if it was here, -70, the energy that you'll use to make it reach -55 is less. But if you are at -80, the energy you need to make it to reach -55 is more. So this period here, the hyperpolarization phase, is a relative refractory period. You can generate an action potential, but you need more energy. More energy is required to generate an action potential during this phase. But during this phase, it is impossible to generate another action potential. These are the properties of action potentials. And so we are now done with the lecture. I don't know whether you have a question. Any question? Okay, those are mine that I've given you. You will answer them at your own time, not a big deal. So we'll now stop there. We are now done with cell biology. So our next series of lectures will be on basic tissues. So during anatomy time, we will look at two basic tissues, epithelial tissues and connective tissues. That is next week. During physiology time, we will look at the excitable tissues. We will look at muscle tissue, and we'll also look at nervous tissue. That is for next week. We'll be looking at basic tissues next week. All right. If there's no question, then I think you'll be free to leave. People are still asking about books. I shared with you people the course outline, and those course outlines have books, reference books within them. You can just check on that. All right, thank you very much. We'll stop there for today.